maybe we can start. First of all, thank you for joining us today for today's webinar. First, I will introduce myself. My name is Amar Tamra. I'm a life science application engineer in Fluigent. And we are very thrilled today to host Dr. Colin Chu from UCL Institute of Ophthalmology in UK. And today he will be sharing with us and guiding us through the IBEX technique that he helped develop with his team in NIH USA.
I'll just share my screen and hopefully, can you all see the presentation now?
Yes, we can.
Wonderful. So thank you again for all giving up your time and coming today. It's really a pleasure, and thank you to Fluigent for organizing this. So I'm going to talk a little bit about the particular approach we used using the Aria machine to do this form of highly multiplexed immunohistochemistry called IBEX that I was privileged enough to be involved with, along with wonderful colleagues during my postdoctoral training at the National Institutes of Health, Dr. Ron Germain's group, and huge thanks to Dr. Andrea Radke, who really led and invented this, and it was a privilege to work with her. So I'm currently a clinician. I actually work in London. I'm an eye specialist working with patients with autoimmune eye disease. But hopefully you'll see why I ended up using this technique and how it will be of relevance to whatever tissue you work on or organ system, and is a very easily accessible and cost-effective way of potentially accessing some of the new exciting technologies that we have available. Before I go any further, the examples I'm going to show you are of the eye, and in particular, I work on a tissue at the back of the eye called the retina. And we're very lucky that because the eye is naturally transparent, we can take photographs such as of the back of the human eye, as you see on the left. But also, the eye is a perfect structure because of its regularity in terms of the retina at the back, that allows us to then know the position of cells and see that. And so it's a really nice example tissue to test these sort of techniques in. And I was interested during my training about learning to access more information, not only from the retina in life, but also in post-mortem samples. And that's what took me to the NIH to work with the team there. And we can do histology on the retina in the same way as you might with your own tissues. And really, we have come a long way, but it's been a long time since Cahal's beautiful hand drawings down the microscope to then H&E staining as shown on the right, that is still the mainstay of most clinical departments using hematoxin and eosin or H&E staining. However, there's a lot that we can't detect from this. And so many of you in a research setting will be doing immunohistochemistry, where we use antibodies targeted to a single protein and then labeled as, for example, here with a fluorescent tag that can then be imaged on it, for example, a confocal or wide field microscope. But we can only really, at least until the last few years, do 1 or 2 or 3 colors at a time. So we can't really see what's going on in the entirety of the tissue. And that's where multiplex imaging comes in. And so the technique I was able to be involved with, developed at NIH, was a technique called IBEX, standing for Iterative Bleaching Extends Multiplexity. And there are many approaches available to you out there now. Some are available commercially, but what we were really focused upon was delivering an open source, straightforward, robust, easy to pick up, and a technique that would work with your existing antibodies by and large. And so this, like many others, you can be done manually, which is what I'm going to show you first, before moving on to show you how we automated this process using some technology from Fluigent. But we would, for example, take a normal slide, coat it with an additional layer of adhesive, section our tissue onto it, and that could be FFPE sections or cryosections. And then would stain with our antibodies with fluorescent markers in this case. And the point I'd want to make here is that we tend to use directly conjugated antibodies in these sort of multiplexing techniques to get around some of the issues of cross-species reactivity. There are ways around that, but by and large in this technique, we did that. You can then stain normally, as many people do, at 4 degrees overnight in the fridge, or you can use some of the technology that we did here, such as a non-heating microwave, to accelerate the staining to around 30 minutes. Because it's flexible enough, we could do it on a confocal or a wide field microscope, inverted or upright. It's really very flexible. Take the image, remove the cover slip, and then the key bit here is bleaching the fluorophores. So this particular technique works by using lithium borohydride, which inactivates almost every fluorophore that we tested. So many of the Alexa fluors, for example, or others. But what's is sort of a handy sort of bug within the system is that nuclear dyes such as DAPI or Hirsch do not bleach, and that can be a good thing because it allows us to keep a fiducial marker to then essentially stain the tissue again with our next set of antibodies and then realign it on the microscope and go round and round. And then at the end, we can use the registration software that was open source again based on Simple ITK that's provided in the initial publications to then just register the images together, essentially to give you a beautiful picture such as this, which is an amazing image from Andrea of a 66 marker antibody IBEX panel on a human mesenteric lymph node. And just zooming in on one follicle, you then realize the depth and richness of this data, because it's almost impossible to show all 66 markers simultaneously, but just showing sort of 3 at a time. Hopefully, even just within one small follicle of a lymph node, you can appreciate the spatial data and complexity that would otherwise be missed just using more normal techniques. And similarly, we've also done work in the retina now in our lab. And so this is, let me just play this. This is human healthy retina, where we just cycle through a single marker. This was done manually on a confocal, but just showing you with a relatively modest number of markers, how this could be any of your tissues of interest, that you could reconstruct almost the entire tissue microenvironment. And that's particularly important here in health to see the layers and the positions of all the different cells, but it's even more critical in many other applications, particularly when you perturb the system. And we're very excited also to then be able to contribute to other consortia. And so many of you may have heard of different initiatives that are mapping all the tissues of the human body, Human Cell Atlas, and here the HubMap initiative that we were very lucky to be able to contribute some of these data sets to as part of their organ mapping antibody panel initiative. And so I'd encourage you all to look at this, because they are developing and maintaining, for example, optimized panels for this sort of multiplex imaging, certainly for IBEX, but also for other approaches you may have. And as I was alluding to, in health, you still get a lot of information, but then as shown at the example on the right here, when things are perturbed, such as during diabetes, so this is what we call diabetic retinopathy, which is one of the leading causes of blindness. Hopefully just, even it's just really a representative example, you can appreciate the sort of complexity, individual cell changes that would be missed without this richness of the number of markers that we're looking at. And it's not just human tissue that IBEX works with, it's very versatile. And so we have a preprint out that you're very welcome to look at with a colleague who works on different model organisms. And so we've been able to show that the fundamental IBEX technique works across many different species, including zebrafish, other rarer types of fish such as killifish, even also to xenopus. And I think this highlights the strength of IBEX, that you can use your existing antibodies without very much modification in this system. Some commercial solutions with fancy sort of oligo tagging, it can be quite difficult to adapt, or you may be tied into, for example, the commercially available ranges, which certainly if you work outside of the very popular, for example, cancer or immunology ranges, you may struggle to access. But for us, using these bespoke and rare markers that, for example, might only be in the eye that we need, we can repurpose and readily integrate these. So that's another advantage. And then this final image is of a mouse retina. So as I mentioned at the start, I work on autoimmune and inflammatory eye disease. And so we run different models, including mouse models of ocular inflammation. And this is just a modest, I think, 10 markers, but showing you a mouse retina during inflammation. And hopefully what you just appreciate is that it's a complete mess and really, really hard to see exactly what's going on, the complexity there, even with these 10 markers, let alone when we then go up to 60. So hopefully just making the point that you could all potentially apply this and benefit from this in your research. But what I've shown you up to this point was all done manually. And so part of the time when I was in NIH, we were looking at ways of increasing the throughput, the efficiency, because, for example, doing it manually, we're taking the slide on and off the microscopes, manually staining and bleaching. There's problems there then with both realignment, but also the sheer amount of time and hands-on time, we call it, as researchers that we're having to put in to generating these data sets. And so that led to this publication, which please do go and read, and again was a wonderful collaboration with Andrea. And we essentially worked by combining different elements from different companies to achieve what we think is a very practical and actually very cost-effective solution, and really facilitated for me as not an engineer but a biologist and researcher through the Aria microfluidic system that enabled us to set this up and interface this very easily. And so I'll talk you through the schematic and zoom into certain parts, but essentially what we did was adapt the IBEX platform and the technology that I've shown you, but to work with an automatic microscopy and microfluidic system that we set up. And we did this by combining essentially elements from 3 different companies. You can, and many of you will be familiar with other commercial solutions where this is all in one. But the part of the reason that certainly my lab in London, we could never afford to really access them is the high platform and startup costs, which often range into the hundreds of thousands. Whereas with this, we were able to get the costs down actually very low or repurpose existing microscope systems. So we were able to take our existing Leica, what we call a thunder imager, which is a type of wide field microscope that can essentially deconvolve wide field images into near confocal like images. And the advantage of that is that it's also camera based, so you can take your images very quickly. So for example, an area of tissue that might take 1 or 2 hours to image on a confocal microscope could be done in the matter of 15 to 20 minutes on the thunder. So that's generally of great use to many of us, but particularly when you might be doing 3, 4, 5, 6, 7, 8 rounds of cycling, all those time savings become really iteratively and exponentially beneficial to you. We'll talk a bit more about Fluigent's Aria, and that really drove the microfluidic element and talking to both the microscope and between it. And then we used these Warner chambers to actually put our sections in and handle the staining. And I'll talk you through essentially how we did this. And so the first part was how do we put our tissue into the system? And because of the microfluidics, we needed to do this in a different way. Essentially, we took cover slips and were able to section our tissue on in the same way as we did before with this adhesive chrome alum gelatin coating that's pretty critical to the technique because it makes sure your tissue doesn't lift off during the many rounds of imaging, washing, staining, and bleaching. In this case, you then have this reservoir, which is around 200 microliters, where the microfluidics would come in from the left, fill it up, would be pushed through into this reservoir and then suctioned away into a waste vacuum pump. This was then clamped together, and this bottom base would not only be heated to 37 degrees, but would remain unmoving on the microscope stage, and then the microscope would be an inverted microscope in this case coming from below to image the tissue. Just to touch on the fact, what we did was we adapted many of our antibodies to do the staining for 1 hour at 37 degrees on the microscope stage. And actually, it was incredibly surprising how many antibodies do work very well using that approach, particularly high affinity monoclonals, ones that you might have adapted from, say, flow cytometry. But you can also extend the length of staining perhaps to 2 or 3 hours for those harder to stain targets. So I'd encourage everyone to think about potentially speeding up your staining workflows. And what does this look like in reality? Well, it's never as beautiful as the schematic. So here is an example of the tissue on a cover slip. We would then fit that on with a little bit of vacuum grease and then insert it, as you can see in B and C, onto the mount. And this is how we would have our tissue located on it. And then the next thing was that we needed all our different solutions, so antibodies, for example, and to be loaded and delivered onto the stage in a controlled manner. And that's where the Fluigent Aria came in. So we have an air compressor leading to the system. The fluid would then come out, go to this what we call 2 switch, which enables us to control waste and then feed our solutions onto the sample on the microscope. We used a heater, and what was really great was that the Aria could connect to the microscope and the software there from the 2 companies, and was very easy for a non-expert engineer such as myself by linking through cables and using this TTL triggering, which is very simple sort of signal out, signal back approach using the 2 types of software, and that enabled us to get them to talk to each other. And so this is how the final sort of setup on the stage looks. We would have the fluid coming from the Aria on the right, filling up the chamber, being sucked out on the right, the fluid coming in on the left, sorry, and then heated to 37. And then this is a picture now of our setup in my lab in London now that we were able to rebuild, hopefully just making the point that actually it's not really a huge footprint system either. You're not going to need a huge amount. You could squeeze this onto a bench. You could almost put it below as well. And these are the different elements. The Aria, as you can see, hopefully here, there are 8 tubes at the front, which are just 15 mil Falcon tubes. And actually, the tubes come right to the bottom, so we can dispense even very small volumes. So, for example, in each of these tubes, we would put the different antibody cocktails for the different rounds of staining in each one, and it would only be around 200 microliters. And that was sufficient to be picked up. We've got these other 2 bottles on the side for what would be our what we call, well, basically for PBS, our running buffer, so to speak, that we would pump through to wash and push our different solutions through. And then essentially inside, the system is basically just a pretty simple pump, which is great. So it's very reliable. It rarely broke for us. And then the software would allow us to talk to the microscope and deliver the solutions through what this 2-way switch, essentially, with one coming in, one to the waste, and one going out to the setup on the microscope. And then this is just an example of the Aria software. So I've mentioned before, I'm not from an engineering background, I don't code. And so for me, it was really important to have a solution that was easy to use. And so the great thing is that it was supplied with the software that's very intuitive. We could almost drag and drop these into the different positions, change the flow rates. We could add our own labels. And so for me as a biologist, I could just pick up and use this graphical user interface approach with the software, rather than having, for example, with some other approaches, having to code or do something more complex. And then this software would readily interface with the microscope software that would wait for what we call a trigger to then bounce back and forth between both the fluidics, essentially doing the staining, then notifying the microscope to commence its imaging. It takes the images and then sends a signal back to tell the Aria to continue to the next stage, where it would bleach, wash, and then stain the next panel, and so forth and so on. Essentially, what this would allow us to do would be to stain 6 to 8 rounds, essentially once you've set it up overnight, and go away and leave it to work. So certainly the students really love this because it can increase their efficiency, so that they then have most of the day free. It's not taken up with taking slides on and off, and also I would argue that in some ways it would make your imaging even more reliable, because the alignment is even easier because you're not taking it off the microscope. And so what sort of images do we get? Well, here are some ones from the paper, really beautiful across a range of different tissues. So this is skin. You've got all the different epithelium and different layers. Hopefully you're appreciating that these look very comparable to the confocal-like images. We're not getting weak staining, despite potentially diluting things as it flows through the flow cell. Here's also in human jejunum and gut, seeing the beautiful villi, and we're able to resolve even down here in cyan to CD3, so the T cells, so single cell resolution of lymphocytes. And here's a lovely movie, again, just iterating through all the different regions you could look at here on a human lymph node. So hopefully if you cast your mind back to the one we showed at the start, which was done on the confocal, really we're now at the stage where we can get very close to that level of staining, but this was all done essentially by setting it going and letting it run by itself for during the day and overnight. So it's sort of the next stage of making things easier and easier, and this was set up really at a relatively low cost. So let's just let that finish playing. And then just as an example of how we're using it, this is from Lorna, a PhD student in my lab. This is an unusual tissue from the eye called the ciliary body that's responsible for producing fluid that keeps the eye pumped up and maintaining pressure. We investigate this from a variety of different aspects and eye diseases. But one question we had was actually to use it in another technique called flow cytometry, where we wanted to identify a surface marker of a particular type of the epithelial cells in the center. And that wasn't known. And so we could then apply IBEX, put a whole range of markers on, and then from this we're able to identify a few such as Vimentin PNA, for example, Connexin 43 that did mark those cells quite specifically in this organ. And so it was just an example of how then the IBEX, beyond its normal applications, also helped us enhance our other workflows. And so I'll begin to wrap up just by also talking about how IBEX, again, driven by Andrew and Ziv's initiative, is really designed to be an open science-based platform. All the protocols and how to do it are freely available. And then on GitHub, you can get it through the QR code or searching online. We have the IBEX Imaging Community, which not only has a range of protocols, links, videos, and the Fluigent team also have many of these links, but also has a repository of both fluorophores that have been tested and known to bleach different tissues, and then over 1,000 different what we call community validated antibodies. So as many of you are familiar, one of the biggest challenges with immunohistochemistry, and that gets even worse if you're doing 60 markers, is finding really high quality antibodies that work. And so this is a great repository, and there are other parallel initiatives, but ones that have been validated to work with IBEX and with the bleaching for a range of tissues, human, mouse, and other model species, and then for many different targets. And these will be validated by individual users who can use their sort of ORCID IDs to then also say, oh, I've similarly found that antibody works, almost a sort of rating or star system. So then you can look at this and determine which ones might also work for you, and everyone can save more time and money by getting straight to the good answers and good science. And I'd encourage you to look at this also for some other videos on how to get started, and certainly a lot more information on how even to set up the 3D automation is available both in the initial publication on the community here, and also Fluigent have a web page that will give you some guidance. And so with that, I'll end and just thank you all for coming and listening. Thank you.
Thank you so much for the very interesting presentation.
Thank you.
If you have any question, please do not hesitate to ask. I have a question. It's in multiple parts, but I'll try to resume it. Someone is asking about a problem in, they have the same system, but he has an issue during incubation due to the vacuum line that dry completely the chamber. So he's asking if you encountered a similar issue. I don't know if he means the system, like the IBEX system, or the Fluigent, the Aria system itself. So I will let you maybe comment and then I will answer.
Sure. Yeah, I wonder if what's happening. So some of these Warner chambers, we had this problem, and it's because you probably saw in some of the images that there's this little loop at the vacuum area. And because the way that Aria works is that it just pushes, it doesn't push and pull, so to speak. So it relies on sort of passive just aspiration of the fluid. And we certainly found sometimes that if you directly connected the vacuum, it would then suck everything out and dry things out. So actually the way the chamber is designed is that you want it to only suck fluid when it fills up above a certain level. So you're never essentially pulling fluid out of the chamber. You're just letting it dribble out passively and then removing it. So certainly what I found is sometimes if the resistance loop got bent, or if your aspiration line was pushed too far in, it would then directly sort of couple to the fluid in the chamber and then suck it out even when there was no flow. So I wonder if that may be it. So I would try and just make sure you pull the vacuum line out a bit of that reservoir, and maybe even reduce, you know, that's probably the main thing. Or if you're using the resistance loop, make sure that the angle is slightly angled up, because then it uses a bit of gravity again to make sure that you don't have this direct connection to the inlet and the inflow. At least the way this works is you're constantly just pushing fluid through. I hope that maybe give some insight and some help.
I hope that was your question, Anthony, and that you have your answer. And if not, if it's related to Aria itself, please, you can contact our support team.
I think there's another, maybe I can come back, there's some more comments from Anthony. Yes, unfortunately, the original chamber, which was sealed from Warner, yes, they then discontinued, which is quite frustrating. So they do have now an open one. So I think what happens then is and I have to say, we will be doing a bit more work on this. We've not yet done it. We've just acquired a new chamber. We're lucky enough to still have our closed ones available. So I think what you would need to do is potentially still cover it, even though it's open, maybe a bit of parafilm so that the evaporation doesn't cause the problem. Obviously, if the chamber's heated to 37, that may also be problematic because it's evaporating. It may be that we have to adapt what we do. So in the original protocol, we would fill the chamber with antibodies and then pause all flow for an hour to allow them to bind. Potentially using a larger volume, but keeping some degree of flow moving may be something we need to iterate with. But yes, I appreciate that. That's frustrating, and I hope we can share your experience through the IBEX GitHub Q&A chat portal, or we can follow up after. So yes, in terms of the antibodies, what I would think is that normally we use quite a concentrated small amount, and you would pause. I agree that if you were to increase the volume and keep the flow going, I would actually significantly dilute the antibodies. So I'd still use the net amount of antibodies, but hopefully as long as it was flowing but at a slower flow, the expectation would be that they would still bind the epitopes as they came past. So you may not necessarily, I would try not necessarily hugely increasing the amount of antibody in the volume, but running a much more dilute solution of it continuously across the tissue but delivering ultimately the same net volume. There may be some optimization there.
Okay, we have another question. The tissue of interest is bone, which is hair tissue and hard tissue and soft tissue combined. As such, we sometimes have a difficult situation. The tissue is being moved detached. If that happens, the whole experiment fails as the alignment does not work.
Yes, thank you. Yeah, so this is it's always very difficult to work with bone and many tissues, the skin as well. They're prone to lifting. It's a little trickier here if you're using cover slips, but coating with the chrome alum gelatin, we found does help. There are many tricks along that line that you do need to coat the slide with the correct amount, not too much. Allow it to dry fully at potentially 60 degrees in a heated oven for an hour before then sectioning, for example, your tissue onto it, and then really allowing it to dry well. So if you can, actually overnight at room temperature before then rehydrating it. That, at least in our experience, seems to give us the best adhesion, but sometimes things do not still lift. The sectioning is really critical, so making sure it's not folded at all in any way, making sure it's been fixed properly, cryopreserved, for example, potentially a smaller thickness as much as is possible, but I appreciate that's tricky with certain types of tissue, would hopefully help. That would probably be where I'd start.
Question. Hello, I'm a postdoc researcher in Barcelona. We are applying IBEX, but in spleen. We also have the same problem in frozen sections. The sample comes up. How can we avoid it?
Yes, so I think again it goes back similar to some of the issues. It's about so if you're doing it manually, then using Superfrost Plus slides that are charged and then applying the chrome alum gelatin, and then those extra tricks about really allowing it to dry fully before sectioning onto it. Try and adjust the thickness if you can, so not too thick. Probably we don't go more than 15 microns in our cryosectioning. Make sure, I know if you have a very large sample, so you might want to, if you can, make it slightly smaller, ensure it has an appropriate sized rim of OCT media around it. That ratio is really important. Also making sure the tissue is not folded at any point. Really the the section, like all these elements, you know, the previous ones, if they're not as good as they should be, they make the subsequent ones even harder. So doing sectioning really flat, being really meticulous about that, and then drying it properly overnight at room temperature, allowing it to completely dry out before you rehydrate. That's really, yeah. So thank you. So 20 microns. If you could try maybe 15, we would normally not go above 15. 12 to 15 can sometimes help. May just be a bit thick. Being very careful during washing and aspiration. Sometimes and again, if you're using a cover slip, don't push the cover slide off. Make sure that you put it in a 50 ml Falcon of PBS and just leave it vertically for the cover slip to spontaneously fall off after about 10-15 minutes. So again, there's a few tricks of not touching them or disturbing it during processing. That may also help. Normally, yeah, I have to say normally, you know, if we can get it to work through the first few cycles, it will keep going. It depends. Ours, I guess, you know, following all those steps, normally for us, it would be pretty rare. Maybe 1 in 10 might lift, and normally not till at least the 5th or 6th cycle. So, and again, I know one other element is that the chrome alum gelatin in our paper is only the liquid form pre-made is only available in the US. And so, but you can make it yourself. There are many recipes online and on the GitHub knowledge base of how to make it yourself from the powders that we can get in Europe, for example, and the rest of the world. And yeah, thank you for the comment. Yeah, the tissue folds. Yes. So that's really critical, because if it does fold, then it's not attached. There's a potential space for air bubbles to form. So the way that the lithium borohydride operates is that when it basically essentially generates a small amount of hydrogen gas through the reaction, and so those tiny bubbles, if there are any gaps in your tissue, can get under the tissue, and then as you bleach, it will lift your tissue off. So potentially, when you do your tissue grossing, cutting the tissue so it's smaller, or whatever manipulations you need to do to make sure that when you section it, it's completely flat. So I guess for you, I would, yes, really focus on the cryosectioning actually rather than the other elements. Make it thinner, about 15. Try and get ones that are either a smaller mold, and so that it's not folded. See if that helps. And I think there was one from another one from Anthony. Yeah.
Yes.
So thank you. So yeah, with Codex. So we don't use the Codex approach, but in theory, I don't see a particular problem with it. It's you're essentially putting the same washes through. Obviously it's different reagents, but then as you say in Codex, you're using oligonucleotide tagged antibodies and fluorophores. We did do a little bit. So in the original PNAS paper, we talk about using total seq antibodies from BioLegend, which are the same sort of principle with an oligonucleotide tag. We showed that manually. We did have 1 or 2 goes with the system, and it would sort of stain and strip. So I think it will work, but we've definitely not spent a lot of time optimizing for that. But personally, I don't think there would be a problem. The only thing I can think of is using slightly more toxic chemicals like formamide. You might not necessarily, you want to be very careful with that. But otherwise, I suspect that a Codex type approach might work. It's just potentially very expensive if you're going to buy oligonucleotide tagged antibodies, and you can do conjugations yourself, but again, it's these extra steps where there's QC issues. So if you can buy, for example, readily commercially available directly conjugated fluorophore antibodies, you know the quality and reproducibility will be a bit easier to control. That's why we stuck at that.
Okay. I have a question, maybe not related to the application itself, but when you put antibodies in the reservoir of the Aria, what is the time that they remain? And is it at room temperature? So no problem for your antibodies to stay at room temperature?
Yes, so yes, so we load them into the Aria machine. We'd normally do 6 or 7 panels, and we leave them at room temperature. Yes, so around roughly 20 degrees. The room we have our microscope in has some air conditioning just so it doesn't get too hot, but around room temperature. Most antibodies are very happy at room temperature or body temperature, right? So so at least, you know, for a day or two, you know. So yeah, we've never seen a problem with that. Yes. And then in terms of the staining on the chamber at 37 for an hour seems to work pretty well.
Yes. Any ways to reduce incubation time?
So I assume you're talking about the staining of the antibodies. So heating, as we do to 37, I wouldn't go much above that. I think an hour at 37 for me is pretty good. We can normally do. I don't think we'd be doing more panels than that over a sort of 24-hour period. The only other way would be, to use like we showed in the initial paper. So we use it's a biowave. It's been basically the technology comes from the electron microscopy field, where they've been using these biowave pros, and I think Ted Peller make one. And essentially what it is it's a microwave, but it has a cooling plate with a cold pump, so it extracts heat. So you can put your slide on that, use the microwave, and essentially, at least as far as I understand it, the microwave causes the water molecules to vibrate very fast, and that accelerates penetration of, for example, your antibodies. And yet you don't get the heating effect because of the cooling exchange mechanism. And so that, I think, is one way of doing it. The trouble is it's pretty hard to put all your microfluidics and a microscope within a microwave chamber. So that's been one issue. Whether there are other chemical approaches we've not explored, but yeah, some good ideas. I think heating seems to work for most of us, and even though I said we do most of them at 1 hour at 37, if you do have some low abundance protein targets or the antibodies aren't great, then what we would do is cluster them all into a one panel and then do, say, 2 or 3 hours at 37, or you could even leave it for longer at room temperature. So normally with a bit of jiggle room, going for a panel of about 30-40 markers, then I think most of these strategies will be good enough. And I'm sure as we go through the future, there'll be even more exciting techniques coming out.
Okay. With heating, is there no problem with unspecific binding?
Yeah, it's a great question. And so that was the commonly held assumption, right? And I think as scientists, I always reflect on the fact that we're very rigorous when it comes to our defined experiments. But when it comes to technical optimization, we often follow what, our elders have taught us, and don't really question these commonly held assumptions. And so certainly we found with high affinity monoclonal antibodies, the staining we see for 1 hour at 37 looks almost identical to that we get at 4 degrees overnight. I think with some polyclonals or very poor quality antibodies, then yes, you do see a little more background. So you probably do want to be a bit selective, but actually, if you have a look and have a go yourself, you'd be surprised at how many work. So most flow cytometry antibodies are ones designed for flow cytometry that work on tissue. We almost invariably find they work very well for 1 hour at 37. So yeah, it's a good point.
Do you perform background subtraction, and how?
Yeah, so I don't routinely. Obviously when so we acquire the images, and you can do different microscopes will have a sort of dye separation feature. So certainly we try and correct for overspill between the channels to a degree. In terms of the background subtraction, we don't do that uniformly, but you can obviously adjust your image when you're looking at it. What we tend to do is more segmentation, would go then to the segmentation, and then when you, for example, pull out features such as MFIs or mean fluorescence intensity, then some of that background might drop out. But at least I have to say our signal to noise for the antibodies we choose to proceed with are often very high, and so we don't specifically do that. But you can in many systems make a baseline measurement and then remove it. Whether that changes across time is always the worry, and between different fluorophores and between different markers. So personally, I don't routinely do it. The background, yeah, so the Thunder background correction, I'm not sure if you mean the ICC deconvolution, or do you mean the Les Link shading? Oh, okay. So yes, we do use the Thunder, what they call ICC, the instantaneous computational clearing. So yes, so we do use that. That is, otherwise your images are sort of out of focus wide field ones. If you do thin paraffin sections, so 4 micron, say, FFPE, it's not necessarily as crucial. But if you're doing a thicker sort of 15 micron cryosection, then yes, you'll get a lot of scatter from the wide field system. And then we use the inbuilt technology. In this instance, in the Thunder, there are different options, but we always use the one called the ICC, which essentially crops what they call the point spread function in a relatively standardized and predictable way. And that eliminates a lot of out of focus light, giving you the crisp near confocal images. So yes, we do that. Yes, it removes background mainly because in this instance, as far as I understand, it's not really background from the tissue, it's background from the light that's above and below your plane of focus that, for example, in a confocal microscope, the pinhole would reject. But in this case, it collects it and then does the calculation to crop that out of focus light out using the deconvolution approach rather than a pinhole. And no, very grateful for the interaction and questions. Thank you, Anthony.
So first question, if he understands properly, immunohistochemistry is realized in situ on the very stage of the microscope. Is that true?
That's right. Sorry, yes, I should explain even more clearly, but that's exactly right. So we put the cover slip on, we leave it on the microscope stage and do not move it from that position throughout the whole imaging process. So that really helps with alignment issues. So when we've done the IBEX manually in the past, we were taking the slide on and off the microscope stage. And while there are tricks you can do, such as aligning it sort of to the top left corner, making sure that's precisely arranged, opening your previous image and then realigning manually, that it was much more challenging. Here, because we can do it all on the stage, there's almost no post-processing or registration required, although we still do a little bit, which again you'd see in the paper uses a simple ITK affine transformation, but actually the tissue, almost barely moves. There is a function you can use in the software called autofocus correction, which can account for any bowing in the cover slip and shift, for example, your Z-stack. But by and large, that isn't required.
Yeah, because we have another question. He's asking about if you can give a comment on the stability of the system. Is the flow introducing drift of the stage?
Yeah, great question. So we have not observed significant drift in XY. So the stage and the inserts are very well designed. They click in very firmly. There are little screws you can tighten down. So that's using the Warner system. So we certainly don't notice any XY drift in any of the time that we were using it. We did occasionally see a sort of slight shift in where our Z-stack position was. So that's why we then used this autofocus correction. So at least in the scope we used, I think the Leica ones have this. You can turn on this function. Essentially, it uses, I think, a 750 nanometer light and total internal reflection off the objective to cover slip interface, and then will shift and accommodate for any bowing that the pressure from the microfluidics might deliver. But I have to say, actually, at the flow rates we're doing, it's not such an issue. And for example, we would pause the flow during the imaging sequence. We would pause and then allow a short time interval, a delay, before then sending the TTL trigger out to the microscope to allow imaging. So hopefully that also allows any distortions during the flow or during the microfluidics flow process to even out. But yes, a great question. Thank you.
Mm-hmm. Another question, isn't there static hindrance around the antigens after a number of cycles?
Yep, great question. I think so. This question gets asked a lot, and I think the answer is that steric hindrance is possible. And I need to go back to the original paper, but certainly Andrea did a lot of testing of many orders, choosing many markers that clustered on the membrane of the same cells. And actually, by and large, it never seems to be a problem. I would say that out of many panels we've done and the testing that she did, I think only in a small handful did it seem to be a problem. You're right in that here, unlike some techniques, we're not stripping stripping the antibodies off, so they are potentially accumulating. But actually, I think in day-to-day with the vast majority of markers, we don't see a significant issue. We didn't mention it here, but you can actually combine stripping with IBEX bleaching. So we've done that now recently, not in the original paper, but you can periodically. So for example, you could do 2 rounds of bleaching and then insert an antibody stripping round and then carry on with the bleaching, and using at least, some standard stripping reagents, it seems to be intact enough. So if you did have a concern around steric hindrance, that's one option. But I have to say, in our hands, for most of the markers we use, particularly if you're very familiar with the patterns and the expected intensities, we never notice an issue. And then in the original publication, Andrea did specifically test that.
Okay. So would it be feasible to use this workflow in a live cell culture or organoids? I have this question also. I was interested in knowing that. Yes.
Yeah, good question. So we didn't set out just because our own experience is working with tissues. So we didn't specifically look at these. However, we have tried it with cells in culture, so sort of immortalized cell cultures like 293 T cells, 3T3s, and it is feasible. We've done, I think, 4 rounds, 5 rounds. The main issue is getting the cells to adhere, which potentially, if you're growing them on the cover slip, may be more tricky. What we found is, if you can spin them down, for example, so immune cells, we can do this thing called cytospin, where we could still coat the cover slip with our chrome gelatin that we know really adheres tissue strongly, so cells in the same way, and you can use a technique to spin immune cells down, for example, and that seemed to work. Growing them on a glass cover slip is also possible, or a chamber, and you could try that, although we've not done that with this automated solution, but it seems to work if you do it manually. In terms of organoids, I've not done it. My worry would be they're larger and the adherence to the cover slip at the bottom would be an issue. However, if you could, if you needed them live. However, we've certainly we've done manually, we've cryosectioned organoids, and then you can adhere them and image them, but not in a necessarily a live way. But there may be other groups out there that have approaches specifically optimized for this. Ours was not targeted to it.
Okay. I have a question. I think it's for me, this one. Is it possible to exchange the flow rate unit from L to M? Yes, it is possible. However, you may need to contact our support team because we might need to come over to change it for you, or to send the Aria back to us to do it in-house, and then we will send it back to you. How would you describe the learning curve to work with the Aria?
Yes, thank you for that. So I think, as I was saying, because I'm a biologist and I've never worked with microfluidics before, I do it very quick. It took me probably about 2 or 3 runs to figure out how all the different parts worked, but it was very intuitive for me as a biologist. So we would, with that software interface I show you, could just change the volume and see, essentially see till it filled our chamber, speed it up, slow it down. It was very, very user friendly. So we went probably from, after 3 or 4 runs, it was sort of doing what we wanted, and then I guess another 5 to 10 more, and then we were almost writing the paper. So I would say the Aria was very straightforward, and we had some good support from the Fluigent team. Very grateful. The tricky part, I think, that can sometimes happen is connecting 2 different devices. So also then you need to think about your microscope. So we were lucky that we also had support from Leica. So in their software, it has a triggering module, which you need, and a way of setting it up. So you need to then get both to be able to work on this TTL signal based interaction. So that took a little more time, working out 2 sets of software and getting them to interact. But I would say very quick with the Aria. I think that was the attraction. We looked at other potential options that were much more sort of modular, or you had to build, or you had to have much more knowledge, or you had to be able to code or program, which was not in our lab's expertise. And so the fact that it had a very nice graphical user interface, so we could just go straight away. So yes, I would say a very shallow learning curve.
We have a question. Is it possible to work with small volumes like 40 microliter?
I think the answer is probably yes.
Yes.
I think so. So, you can adjust it to dispense very small quantities. There's obviously, depending what you're filling, it could be quite tricky, but I think you can definitely dispense volumes. We were working with filling our chamber with 200 microliters. So I think 40 would be very easy. What you would do is prime all the lines right up to whatever device you're using. And then, yes, then it could just do that. You'd set the flow rate and the certain amount of time. And I think the flow rate can go very low,
Yes,
Amar.
it can go very low, but I need to clarify one thing. The volume that needs to be put in the tubes, in the tubings, I think the minimum is, if I'm not mistaken, 40 microliter. So we cannot go below that in order for us to prime the whole system and then to dispense and not to inject air. But yes, I would say the minimum is 40. We cannot go down.
Yes, sorry. Yeah, to clarify, so all the lines and the tubes, for example, we would prime with PBS. So yes, you do have a much larger volume, for example, so in your lines. And so if,
Oh,
for
okay.
example, you were thinking about the antibodies, like this might be, I'm
Yes.
assuming between the
Yes.
question, we want to use as little as possible. So our
Exactly.
antibodies, we were dispensing 200 microliters of the actual antibody, but that was then pushed through the lines all the way to the
It's
chamber
PBS.
with PBS.
Yes.
And so I think sometimes people thought, would that dilute it out in the tubing? But actually, we didn't see that. It seemed to work pretty well for us. So,
Mm.
so yes, I guess depending on how I'm reading the question, I think
Yeah.
if it was 40 microliters of antibodies, you could probably do that, but you would need some running buffer to line the tubing, as you said, Amal. Yeah.
Yes. Yes. Okay. I had a question, and I think you already mentioned it, but I just wanted to be like clear. If we compare your IBEX traditional method of doing it, you would spend like how much time would you spend compared to if we connected to the microfluidics and we connected to Aria?
Yes. So so if we were doing it on a slide, what we traditionally do would put it on and off the microscope, and then you have to remove it to
Yes.
bleach and then to stain your next panel. So if you were staining overnight, which is maybe not the fair comparison, but say doing 1 hour at 37, you would still need to put it back on the microscope and then do the next run. So realistically, with all the manual handling steps, washing and gaps in between, I don't think my students would do more than 2 panels a day. Whereas, as I said, with the 6 tubes, you could do 6 panels in one go overnight. So you're using time that's not available. What we've now started doing is, obviously there are only 10 positions. So we do 6 panels. We start in the morning, run 6 panels, and then just before we go home, we then change the 6 for a different set. And so we can do 12 very easily as well overnight over a 24-hour period. So it is faster. I think actually for me, it's not even the speed, it's just the hands-on time is really low. And even though many PIs, we think our students have lots of time, they can be doing much better things with that. And then, for example, the quality is better because the risk of, for example, dropping the slide drying out, washing the cover slips off, any other potential things that could go wrong at any point are mitigated.
Okay, we have another question. Thank you for the nice presentation. You connected the Aria system to the microscope and automated perfusion steps and microscopy image steps in the same protocol. Is that right?
Yes, that is correct. Sorry, yeah, I could clarify that. So we got the 2 softwares to talk to each other. So the Aria we would start the Aria by washing the OCT off our cryosection and then doing a DAPI stain and then blocking all on the Aria. Then we would stain our first set of antibodies and then we would wash and then we would then the Aria software would then send the trigger to the microscope, which we had set up. It would then trigger, acquire the region that we wanted, and then the microscope would send the signal back to the Aria. The Aria would then proceed through the next steps in that workflow, which would be to bleach and then wash and then add the next set of antibodies and so on and so forth. So you set up the 2 together, the Aria and the microscope, you click go, and then when you come back the next day, you essentially have all your microscope files, sort of the panels all there ready to go. So the only thing you then need to do is take those files, potentially run them through the registration software if you just wanted to really enhance any sort of X, Y, or Z registration, but otherwise you could then put it straight into your analysis software.
Okay, thank you. The system then knows where the tubing volume ends and then the sample starts.
I think I can quickly talk, but I think that's also one from you, Amma. So I think you can define, so when you start by priming the system, so then you will naturally, as you set it up, measure the length of tubing that you need to get it from your Aria to your microscope stage. So you manually determine that and cut the tubing. And then the very first step when you set it up is that it would push fluid through and you say stop when the fluid comes out the end. So then the system knows what the dead space, so to speak, inside the system is. Then anything that it adds that you program in, for example, is meant to be for your sample. So then, yes, it would be calculated at the end of the tubing. So it was quite good. We knew that our chamber held around 200 and we could type literally 200 microliters in and that would fill it. So it was pretty intuitive in that sense.
Because if I might add something, at the beginning of any experiment, there is a calibration step that should be done in order for the system to know the internal volume of your tubings up until, so from the tip of the tubing inside the reservoir until the tip of the tubing that goes into your sample. So it is calculated with this calibration step. And in the meantime, I want also to remind you that if anyone has any question for Fluigent or for Dr. Colin Chu, you can contact us via contact for Fluigent. And if you need to ask Dr. Chu afterwards, we can put you in contact with him if you have a question regarding the protocol itself. I want to thank you again so much. The webinar is very, very exciting. And thank you for the people who joined. And yeah, bye.
Great. Thank you, everyone. Goodbye.